I got the signal from above that we can begin. So welcome, everybody. It's great to see everybody here. And we were just talking, it's a great turnout in person, which is always nice to see. But we are also very fortunate to have those of us who are joining us online as well. So thank you all for being here. As you know, this is a series that gives us a chance to celebrate and to hear from our newly appointed and promoted professors, as well as our senior scientists in the school. And today, I am thrilled that we get to hear from Jennifer Schrack, a professor of epidemiology and also the director of the longstanding Center on Aging and Health. It's always a special treat when the speaker is also an alum of the school, as is the case with Jennifer. Jennifer received her PhD from the Bloomberg School, and even at that very early stage in her career, her work on mobility was groundbreaking. Today, she is a world-renowned researcher who studies the connection between measurement and movement and health, leading some of the most important studies in our field. More specifically, she is a pioneer in working with wearable monitors and is leading the way on developing the proper methods and principles for using these devices. At the heart of all of this work, Jennifer is helping us to imagine a future where more people can stay independent as they age, a goal that grows more urgent every day. By 2035, which really is just around the corner, older adults will outnumber the young for the first time in human history. The changes and the challenges that come with this demographic shift are enormous, but so are the opportunities. In the years to come, if we can help people everywhere to live longer and healthier lives, we will benefit from what older people can bring to our society. And I guess I'm an older adult these days. So as an older adult, I look forward to that brighter future. Of course, the work ahead is quite complex, but Jennifer and her colleagues at the Bloomberg School are pursuing some incredible ideas and innovations in this area. Jennifer has made notable contributions to modernizing our research efforts when it comes to the design, collection, and analysis of data from wearable technology. Working in close partnerships with faculty in the Department of Biostatistics, including Ciprian Crenechano. Is Ciprian here? He may be online. I hope I didn't butcher his name too badly. Good. I've known Ciprian for a long time, but it's always Ciprian, right? Yeah. It's kind of an icon, Ciprian. He just has one name, Ciprian. Yeah. But together with Ciprian and others in biostatistics, she has established the infrastructure for incorporating new wearable and implantable technologies into existing studies. And these includes ERIC, NHATS, PASOS, and that's a lot of acronyms, I know, but behind all those letters is revolutionary work to gather and analyze an extraordinary amount of data. All of those data are helping us to better understand how we are aging. With this wealth of information, Jennifer is shedding new light on late life disability trends and showing the remarkably strong links between physical activity and health outcomes later in life. And just 2 months ago, our school shared a remarkable study led by, and here's another name I could just butcher, but Amal Wanagatunga. Yeah. Oh, okay. Yeah. Amal led the study. Jennifer was a co-author of the study. And this study showed that just 35 minutes of moderate to vigorous exercise every week is associated with a 41% reduction in dementia risk. That's really important information. That kind of knowledge has power and promise as we look to build a better future for people everywhere as they age. Now, Jennifer is helping us to work toward that future, not only through her research, but also by nurturing those around her. She is a much-loved mentor who cares deeply about the next generation of researchers. She is known for the time and attention she gives to promoting their visibility and connections, as well as developing both their expertise and career skills. And I think looking at the number of students and early career faculty in the audience and online is a testament to that. Jennifer has also emerged as an outstanding leader of our Center for Aging and Health, which you just took over, what?
It's been almost 2 years.
2 years, almost 2 years. Seems like just yesterday.
Yeah.
But in that short amount of time, she really has re-energized connections among its members and with the broader university community. And she is generating visibility for the center and for aging science here at Hopkins at a whole new level. And I know she is just getting started. As Jennifer wrote in a recent issue of our school's magazine, we need to recognize life is short and every day that we age is a gift. Her advice to us all is the following, and I quote, "Keep moving, stay engaged, socially and cognitively. Live life with purpose and meaning. Take time to breathe, grieve, and seek joy in what matters most." And I think those words are more important than ever. So take heart. I am so grateful to Jennifer for all the work that she's done to ensure that more people pursue these goals and enjoy the gift of aging as long as possible. So Jennifer, thank you for all you do for our school and for the field of public health, and we are so excited to hear from you today.
Well, thank you.
The floor is
Thank you
yours.
so much. Great. Thank you. Thanks, everyone. Thank you so much. Thank you, Dee McKenzie. That was a lovely introduction. And thank you, everyone, for coming. I know that this is a tough time for public health, and a lot of us have taken a beating over the last 100 days. So it really means a lot to see so many people here and supporting. I have to say there was a time about a month ago where I wasn't even sure I wanted to do this after another round of bad news came in. But I think if we stop doing this, then the bad guys win. So I'm here, and with that, I think that we can keep going. So let's talk about physical activity and aging. So I want to talk a little bit about why and how I got interested in this, because I didn't take your typical career path. Actually, my undergraduate degree is in accounting. I don't even have that up here, but I went to Villanova University and I majored in accounting because I didn't know what I wanted to do. I liked numbers, and looking back now, it makes a lot of sense. My favorite classes as an undergrad were statistics and calculus. And my advisor at the time said, well, why don't you think about accounting? There's a lot of jobs and it's a good quantitative field. And so I took the classes, and the classes weren't bad. They're very logical. They have what they call projections, which is what we do as prediction modeling. It's actually, there's a lot of similarities, but the outcomes are different. And so I did that for about 5 years because I needed the money. But I also didn't know what I wanted to do. I had always enjoyed health and fitness. I was on a lot of sports as a kid. I enjoyed reading about health and fitness, but I'd never thought about it as a career. But I took some night classes at community college, anatomy, physiology, nutrition, and applied to a master's program in kinesiology at University of Michigan because I was living in Ann Arbor at the time. And I loved it. I loved every minute of the 2 years that I was there. I went into that program thinking I was going to have a life in cardiovascular fitness, weight management, metabolic disorders. That was kind of my interest at that time. But while I was there, I was asked to help with a study that had to do with physical activity and falls in older adults. So working with a leader in geriatrics out at Michigan, his name is Neil Alexander, he asked me to help work basically data collection. I was a master's level person at that time, help with strength training. So it was training the hip flexors and the quadricep muscles in the lower legs to help prevent falls. And so it was a small pilot study, but I was really fascinated by it because it was something so simple. These people came in twice a week and they exercised, and it made a huge difference in their quality of life. They felt, just talking with the participants, they felt their agility increase. They just felt better. They enjoyed the engagement. They'd bring me recipes. They'd bring me food. Like, it was just really fun. I really enjoyed it. And it made me want to study aging. So that led me to a job at the National Institute on Aging. Does the pointer work here? There we go. So this is me back in 2003. I got a job working at the NIH on the Baltimore Longitudinal Study of Aging, which is here in Baltimore. It's run out of Harbor Hospital on the south side of Federal Hill. And these are pictures that are taken for a brochure, and some of them are still on their website if you go there. So that's not a real participant, that's someone else who works there, but that's me showing how to do a lower extremity strength test. On the far left and on the right, I'm doing a treadmill test for cardiovascular fitness and fatigue. And I really liked that. And the BLSA, I still work with them today. I wrote my whole thesis in BLSA data. My heart it has a strong place in my heart, the BLSA. But that motivated me to want to do epidemiology because it was about studying people for life to try to understand who ages well and why. And I thought that was really fascinating. So that led me to do my PhD here, epidemiology of aging. I finished in 2011, did a postdoc at NIA for a year, and came back here as faculty in 2012, and the rest is history. But because of my background in kinesiology, I've always really been interested in movement, in the way people move, in the way they walk. I was reading a Michael Connolly book, and the detective in that book is talking about how everybody has their own way of walking. And I was like, yes, exactly, that's what I'm talking about. Everybody walks in a distinct pattern. It's like a footprint. So I'm fascinated by how people walk. And while I was studying it during my thesis, Luigi Ferrucci was my mentor for my thesis and still is my scientific mentor to this day. He said it crosses species, right? This is not a choice that people make to move less and move slower as they get older because it crosses species. And so I dug into the literature and he's right. Worms, fish, mice, they all move slower as they get older. If you have a dog and you've ever walked that dog as they get older, you know how they move slower. And of course, people move slower as they get older. So what is it? Why do we move slower? And it is probably the most consistent change in physical function is how fast people walk, walking speed or gait speed. So throughout life and up until about age 60, people walk between 1.1 and 1.3 meters per second. A lot of that is dictated by leg length, a little bit by personality, but it tends to be in that range. And Alden and I, when we teach epi of aging, we have a lab class where we put tape on the floor and we clock people's gait speeds, and about 99% of them are in that range. But around age 60, things start to change. And by age 80, we tend to see people walking less than 1 meter per second. And when people get into this range, it's been shown to predict disability and mortality, frailty, and then there's this category called dysmobility, which is less than 0.6 meters per second, which is really quite slow. So people do slow down, but it's not universal. So this is data from the BLSA. This is anyone who's ever had a gait speed test in BLSA. So there's quite a bit of data here. And you can see there's a lot of scatter here and it's color coded. So the light blue is what we consider normal speed. The medium blue is considered kind of intermediate. And then the dark blue is considered slow. And so you see most of the cloud of dark blue is over the older people, but not everybody who's in the older range walks slowly. So that's what we call heterogeneity, right, in public health. So what is it about some of these people who are over the age of 80, over the age of 90, and still walking at what we would consider a normal speed? And so that has long been a fascination of mine, is trying to understand it. And there are a lot of tests that we have traditionally used to understand how people move and how much they move. All of these are clinical laboratory tests. So walking on a treadmill at a certain speed, walking over the floor between two cones at a certain speed, walking as fast as you can, getting up, turning around and coming back and sitting down again. All of these are tests of mobility and walking. There's even a gait lab. This is at the BLSA. They put these little balls all over you, and you can look at not just how fast people walk, but their gait characteristics and how they move, if it's stable, if it's not. But the problem with all of these things is that they are measured in the clinic or in the lab. They're not measured in the person's own environment, where they function, where they live, while they're walking down the street. And so when I test, or any of us test this kind of function, we're getting somebody's optimal performance. There's no distractions, there's no cell phones, there's no pets, there's no toys to trip over, there's no carpeting usually, although you can see there's carpeting in this one. So it's optimal conditions. And so this is why it's so hard to see a relationship between something like these measures and falls, is because this isn't how we function. So there's long been this desire to understand function in people's own environment to a better extent. And so that led to my interest in these. And so I think everybody knows what they are. A lot of us have them. Some of you might even be wearing one. Even if you don't have one, if you hate them, there's one in your phone. They are tracking you, right? And they're tracking how much you move, how fast you move, yes, to a certain extent. There's a lot of measurement error in there, but that's a whole nother lecture that we could give at another time. But they are out there and they're here to stay and they're wildly popular. And so what you see on the left side is more consumer grade. These are the ones that you can go out and buy or order from Amazon and have your own. In the middle is kind of, again, your phone. Down at the bottom is the Oura ring. That's kind of a newer thing. And then on the top is an old school pedometer, the red one up here. These have been around forever. And that really truly does just click up and down each time you step. And then over here on the right side of the screen is what we call research grade accelerometers. And these are more of what we use in research. And they just you have more control over them, and you can program them to collect certain things and turn on and off at certain times. So researchers tend to like those a little bit better. So like I said, they're here to stay. They're incredibly profitable for companies. So this is just looking at the sensors wearable market, where we were. I pulled this in 2024. So that's where we were then, and we're projected to be in terms of global sales of these things by 2032. So I think we as public health researchers need to try to understand what the data mean that come off of these and how we can use this to understand people's health. So this is the consumer side, but in research, we've also seen things take off as well. So these have been around for a while. Of course, the research grade ones back in the 90s and early 2000s. I remember at Michigan wearing one of these. It was about this big and it came in a fanny pack, and it was called a mini logger. And I was running around campus with it. It was not cool because it was a fanny pack, and it was not comfortable either. But you can see this is a PubMed search just of accelerometers and aging research, and it's really taken off. This is a review we published in 2018, so it's actually outdated already. If you extend this, it's certainly even more so. So they are really taken off scientifically as well. And so they really gained popularity. You can see somewhere around here, around 2010 or so. And what this is NHANES put accelerometers in between 2003 and 2004. And that's when it took off, because when that data became publicly available, a lot of people started to look at it. And so this is what that data actually looks like. So any of you have a device, this is what that data would look like if we plotted it on the raw data on a graph and actually looked at it. So this is a snapshot of NHANES data. And back then they wore this red, well, it wasn't the exact red one, but it was like this, and it was worn on a belt at the waist. It wasn't a wrist-worn device. So they were told to wear it during waking hours. So you see between midnight and about 8 a.m., there's no data. And that's because they take it off. They took it off to sleep. But then at about 8 a.m., people get up and they go about their day, and you see these different spikes in the data. And so the higher the spike, the faster the person's moving, or the more vigorously the person's moving. And so what early researchers did with this data, they were really interested. It was mainly physical activity researchers wanted to know about compliance with public health recommendations. So they would draw lines through it. And anything that was between that like magenta colored line and the blue line was considered vigorous activity because it was more intense, more acceleration. Anything in these two lines was considered moderate intensity activity, and anything below that was considered light intensity activity. So basically took all this data, just drew lines through it, and categorized minutes per day spent in moderate to vigorous physical activity to see if people were meeting the physical activity guidelines. Does anybody know what the physical activity guidelines are? Amal's not allowed to answer. Does anybody know what the physical activity guidelines are from the CDC? How much activity are you supposed to get per week? It's roughly 30 minutes a day. It's like 150 minutes a week, roughly 30 minutes a day of moderate or greater intensity activity. The problem is most people don't know what that means, what moderate or greater intensity activity is. They don't know how to determine if they're moving at that rate and how to quantify it. And so some of the first papers that came out of this is one of the most popular papers ever written with accelerometry of physical activity data. And it was a feat in itself because it took data from over 6,000 people and tried to summarize it and understand people's compliance with the recommendations. And the conclusion was that among adults, adherence to the recommendation to obtain 30 minutes a day of physical activity is less than 5%. So looking at that, it's like, wow, we're really sedentary as a nation. And the story got worse when you looked at it by age category. So this is just looking at bouts of activity across different ages. And you can see I've drawn the red box around 60 plus. It said that older adults, only 2.5% of them met the physical activity guidelines. And I looked at that and I thought, so I was a student when this came out, and I thought, what does that mean? Are we really that sedentary, right? Or do we have a measurement problem? Are we not capturing something quite right? So there, I highlighted that 2.4% was overall. So it just didn't make sense to me, because even though they're not exercising, because that's really what this is capturing, it doesn't mean they don't move. And physical activity is supposed to be all movement above resting. So you're all sitting now, you're all resting. When you get up and out of here and go for, walk to your next place, that counts as physical activity, right? All movement above resting. But what you're really measuring here is exercise. So they're not the same thing. So back to the drawing board, back to this data. And I found myself wondering, what about all this data that we ignore, right? So we drew lines through the data to get those numbers, those minutes per day, but there's a lot of spikes in here. There's a lot of times where there's no data. What does all of that mean? And so about this time, I started working with the BLSA data set. And I started working with Vadim and Ciprian and others. And this is one of the first papers that we published together. And all we did, instead of just drawing lines through the data, was start to just look at the data to see what the data would tell us. So this is really simple. All we did here was summarize, on average, how much activity does each person have per day that wore this device? It was a chest-worn device that was stuck to them with electrodes. So it measured physical activity and heart rate. And so the first thing you'll notice, and hopefully you can see, is there's a lot of shading on this slide. And that's because there's a line for each person. There's 611 people in this data set. And there's a ton of variability in what people do. Some people are way high, some people are way low, some people are in the middle. And then we put in 4 data-driven age groups to try to get a handle on what the data looked like. And so what you see here is the green line's less than 60. I think the average age in that group is about 50. Red is 60 to 67, blue is 68 to 74, and then the yellow line is 75 and older. So what we see is, not surprising, that the older people are less active than the younger people. I don't think that's news to anyone. But if you average this out, this is cross-sectional data, the average is about a 1.3% drop in activity per each 1-year increase in age. So what that means is that between the ages of 50 and about 75, you lose about 50% or so of your daily activity. And so that's a lot. That's a lot of activity lost in that age range. We also took the data and looked at it by time of day. So this is the same paper, same people, same data. The only difference is, instead of looking at total activity per day, we're looking at the amount of activity per minute in each minute of the day. So what you see here is from midnight till about 7 a.m., people were sleeping, right? They wore this at all times. And then people got up, went about their day, came home and went to bed. And you see the 2 younger age groups, the lines look pretty much the same, the shape. And then the 2 older age groups, the lines look very similar. But the shape it changes dramatically between 60, 67, and 68 to 74. There's a dramatic shift in the way that activity looks. And does anybody want to take a guess what that is? Retirement. Yeah. So this is cross-sectional, so we can't say people retired and then became less active. But on average, people who were more active or who were employed were more active. So you should never retire because it's bad for you. But there's other things going on here. And we started to pick this apart, try to understand, okay, so it could be lifestyle, could be a shift in lifestyle like retirement, but what about health? And this was about the time that Amal started as postdoc. He started in 2017 as a postdoc with me, and I convinced him to stick around. He's still here 8 years later. And this was one of our first papers, I think, together, looking at the effect of fatigue on daily activity. And so again, this is the same kind of thing. You're going to see a lot of these different plots where it's what we call a diurnal plot or a circadian rhythm. So from midnight to about 6 a.m., again, people are in bed, so we don't see much. And then they get up, go about their day, come home and go to bed. And so we saw that people with higher fatigue were less active. And again, I don't think this was groundbreaking. I don't think it surprised anybody, but it did put a number on it. And fatigue is a huge problem for older people. It's a problem for a lot of people with disease, but it's very nonspecific, right? You don't always know why. Why are people fatigued? Why are they tired? There could be many, many reasons. It could be as simple as you didn't sleep well last night, or it could be that you have 5 chronic conditions and they're not well managed. So we wanted to pick it apart a little bit more, specifically understanding how this was associated with diseases. So this was one early on that we did, was looking at it by cancer status. And I really like this paper. I think this is really, really interesting because right about this time, we were looking at accelerated aging and cancer. So when people get sick with something like cancer, we've also looked at this with HIV, you have to take the treatment or you're going to die. But those treatments can be toxic and they can lead to what people think of as accelerated aging. And so this we looked at, this was in the BLSA, and these people were healthy survivors. And all we did was stratify by cancer history versus no cancer history. And about 30% of the people had a history of cancer. I don't have the N on here, but you can see those people, the red line, those with a history of cancer were less active. And they started to tail off earlier in the day. And when we actually modeled this, what we saw was that the difference in activity was equivalent to about 5 years of age. So what does that mean? That means a 75-year-old with with a history of cancer looks more like an 80-year-old without a history of cancer in terms of their activity patterns. So they're moving slower and they're moving less. And I thought that's a pretty big statement in terms of what the treatment may actually cost you. We also started looking at it by common chronic conditions that older adults have. And one of these was vision loss, so sensory loss. Sensory loss has become a really big deal here at Hopkins, thanks in part to the work of Frank Lin. This was led by Yuran Kai, and she used to be a postdoc of mine. She's now at University of Pittsburgh, and this was also in the BLSA. We were looking at sensory and motor combinations, and this was looking at vision loss. And again, we have the red are people with visual impairment, the blue are people without visual impairment, and we see that those with visual impairment were less active. And again, not surprising, but the thing that is interesting here is that we see the difference pop out more with visual field impairment than we do with visual acuity. So visual acuity is the more common test, though. This is the eye chart. When you go to the eye doctor, visual acuity is what they measure. They rarely measure visual fields unless they have a reason to do so. And so the more common test actually seems to be less sensitive in detecting change in the way that people move and potentially the way people function. And this could be quite important if we're trying to understand something like people's falls risk. We also looked at this in Achieve, this little study called Achieve, which some of you may have heard about. Understanding physical activity and hearing loss was not the primary goal of Achieve. Achieve was about understanding hearing loss and cognition. But I said to Frank, I think we need to understand how much people move. If you have people, you give them hearing aids, right? So that was the object of Achieve, was giving people hearing aids to see if it would slow their cognitive decline. It may actually influence them to move more if they can hear better, if they're more comfortable with their surroundings, if they may become more socially engaged because they can hear better. Will it do anything? So we looked at baseline, and what it was is everybody in Achieve had some hearing loss. The blue line is people with mild hearing impairment, and then the red line is moderate or greater. And we see that those with more hearing loss had less physical activity at baseline, and they also had, particularly in the afternoon hours, as well as poorer balance. And so the important thing about this was, if you look at it just together, if you summarize it into one number and say that on average, here's how many steps per day they had or how many active minutes per day they had, there really wasn't a difference. But if you looked at it over the course of the day, we saw that there was less activity in the afternoon. And interestingly, what happened over time in Achieve was no difference. So the intervention versus the control, after over 3 years, we saw no difference with the intervention group. So the hearing intervention did not attenuate the decline in physical activity over 3 years. We did see a slight difference in cognitive decline within Achieve, the main results, but it did not seem to affect how active people were, how they slept, or how they functioned physically. So that was interesting. It could be that maybe 3 years just isn't enough time. Maybe we need more than 3 years, but Achieve is ongoing for follow-up. So we'll see if this slows things going forward. The other thing we've been really interested in is understanding cognition. So the brain and mobility is a big area, and understanding early indicators of loss in cognition is a high priority for NIH. And so Amal and I did this work in 2022. This is in BLSA, where we grouped people by mild cognitive impairment and Alzheimer's disease versus normal cognition. And we saw again that those with normal cognition had more activity throughout most of the day. Importantly, when Amal looked at this and modeled it all together, because the people with MCI and AD, you can't see it as well, but it tailed off slower at the end of the day. And so they tended to be a little more active later in the evening and in the overnight hours. If you just looked at it again as one number, it didn't matter. But when you looked at it and parsed it out by time of day, there were quite important and significant differences in terms of their cognition. And so this was important. We wanted to understand this a little bit better. And so we started to dig into some of the underlying pathology there. And what we saw, this was led by Francesca Marino, who's a student who graduated last year, was looking at this with Alzheimer's biomarkers and pathology. And we saw that those with A-beta, who are A-beta positive, which is a marker of pathology in the brain, those who were A-beta positive were less active, particularly in the late afternoon and then in the overnight hours. But those who were ApoE carriers, which is the allele that is associated with increased risk of Alzheimer's disease, they were actually more active later in the day and also into the night, which could be a sign of poor sleep. Adam Spira would tell me that that is poor sleep. But this is interesting because the effects seem to oppose each other. And so the question is, do these pathologies work differently? Do they interact differently with mobility? Are they in different parts of the brain? And we don't know yet. This was all cross-sectional data, and it's something that deserves follow-up, but it is a very curious finding. And then Anise, who's here as well, led this paper recently to look at dementia status in NHATS, the National Health and Aging Trend Study. And so we saw a very similar curve where we see people with possible or probable dementia are less active. But the interesting thing here is that possible and probable dementia look very much the same. And that matters because a possible diagnosis or a possible dementia classification is quite different than a probable dementia classification. A probable is a much stricter criteria than possible, but yet their physical activity looks the same. So this makes us wonder, is it possible that the differences in the way people move may emerge in the prodromal stage of Alzheimer's disease or dementia? And could it possibly be a warning indicator? People start to move less, start to move differently, start to move at different times of day. And this we've seen across studies is they tend to extend later in the day, and they're more active later into the evening than we would expect them to be given the shape of their curve. And so this made us wonder about other patterns as well. If you can see here, the red line especially is a little bit wiggly. There seems to be a little bit more variability. Some of that has to do with the sample size. But is there more variability or more something different about the way they move throughout the rest of the day? So if we went back to this data that I showed you at the beginning, where we started, where we were understanding classification of activity, we're thinking about time of day activity. What about the spikes? What do the spikes mean? So if we have a spike and then it's followed by a time where there's no activity, does that mean something? Does that matter? And so we started thinking about these transitions. And so this is where Vadim comes into play. And we started saying, how can we think about this activity? How can we model this activity and think about it? Are you moving, yes or no? And so Vadim had this idea of borrowing from the sleep literature. A lot of sleep researchers like to look at what's called sleep fragmentation. So how broken up is your sleep versus how continuous, how well you sleep. And so we started thinking about how fragmented people's activity was during the day. When you're active, are you likely to stay active or are you more likely to stop and rest? And so that's a lot to wrap your head around. So I have this slide that kind of illustrates it. So if we think about as people get older, so we have the younger person on top and then a middle-aged person and then an older person. And we ask each person to be active for about 10 minutes. This person can easily walk for 10 minutes without stopping. The next person might walk for 8 minutes, stop and rest, and then walk for 2 minutes. And then the older person who may have some mobility challenges might walk for 5 minutes, stop and rest, 3 minutes, stop and rest, and then 2 more minutes. Everybody's accumulating 10 minutes of activity, but in a very different way. Does that matter? Is that important? What does that tell us about health? What does it tell us about their brains? What does it tell us about their function? They have more transitions between active and sedentary states. So this is just a 10-minute window example. But if you extend that across the day, across the week, that they wear it every minute of every day, well, what does that tell us? And one of the first things we looked at with activity, what we call fragmentation or how broken up their activity is, was survival. And so Amal looked at this in the BLSA, and he looked and saw that over up to 10 years of follow-up, for every 10th of a unit higher in their fragmentation, there was a 49% greater mortality risk. So the hazard ratio was 1.49. And the interesting thing about this is that it outperformed total daily activity. So just looking at how much activity they accumulated during the day didn't matter. What mattered was this pattern. How broken up was their activity? How much did they have to stop and rest? What was their probability of transitioning to a sedentary state after they've already been active? That mattered more than just looking at how active people were. So this intrigued us even more, and actually was repeated rather recently by Mauro Mediano, who was a fellow who was working with CUNY when he was here, I think about a year ago. And he published this paper looking at activity patterns and survival in the study of Latinos. So this is a younger population, a very, very different population from what we've seen in BLSA. And we see a very similar result. Now, The risk of death is only 22% versus here it was 49%, but these people are much younger, but it's still there and it's very consistent. And we see the same thing in terms of looking at it by tertiles, the lowest versus the highest. Those who have the most fragmented patterns have a 45% higher risk of death. So given that, what could this tell us about people's brains and what's going on in terms of their memory and their cognition? So Anise looked at this as well in NHATS. And what we saw was, first off, those with dementia were less active. So you can see that here. Sorry, I'm having trouble with this. And they had lower intensity. So on average, they were about 57 minutes per day less active, people with dementia. And again, not that surprising, but their fragmentation index was 5% higher, which is quite a bit, almost 6% higher. So their patterns were quite different. We also looked at this by domain of executive function, orientation, and memory. So these are cognitive performance domains. And those who had higher cognitive performance had were more active. They had more active minutes per day. They had higher intensity per day. They had less fragmentation, so their activity was less broken up. And and very interestingly, they had less variable, more stable activity patterns. So thinking about people get up, when they get up, how active they are, when they go to bed, that was much more stable among people who had better orientation. And orientation is a cognitive domain, and the way we assess it in NHATS is asking people about the day of the week, the date, the month, the year, and then we also ask them who's the president, who's the vice president. So people who were better in terms of their orientation to time and place had better brain function. And so this might seem a little odd to some people because what we're doing is looking at people's cognitive function and looking at how their brains perform, right? It's usually looking at the cognitive function, looking at their physical activity. We usually think about the other way in terms of their physical activity predicting their cognitive performance. Is physical activity, is exercise preventative for poor health outcomes? But I think we know that. I think that, yes, we know that physical activity is good for you. I don't think anybody in here is going to argue that. So we're trying to think beyond that. We're trying to break through that and think about with these sensors where we're really monitoring how much people move all day, every day. What more can we learn from them, right? We're learning more than just exercise is good for you. We're learning about how much people move, how they sleep, how well they sleep, and what that can tell us about their health. So Dean McKenzie mentioned this at the beginning, but we have a lot of projects that we're doing this in. And we're just starting to pool some of this data together. So I mentioned Achieve. That was Frank's trial on cognition and hearing aids. Eric, the neurocognitive study, we're monitoring people in that study. The BLSA, of course, we're still working with that. We did do it in the MAX at one time point. NHATS, we have about 1,000 people that we've monitored. We're going to blow that up and do everybody, hopefully next summer. So that'll be over 6,000 people. PASOS, which is part of the study of Latinos, so Cooney's the PI of that study. We have about 4,000 or close to 4,000 people in that one. We did a small study of physical resilience in older adults. That was about 300. And then STURDY was a study of vitamin D in falls. And we did monitoring on 688 people. So we've done a lot of work collecting a lot of data. So now what? So we've kind of just, I think, scratched the surface and understanding more about what these activity patterns can tell us. So what more can we do? So we've started trying to think about how we can combine some of these cohorts to really leverage a lot of this data and do even bigger studies. So we're going to take a deeper dive, first off, into physical activity and frailty. And so this is work that Lacey Etzkorn's helping me with. Lacey is a graduate of the Biostats program here, and now I'm very happy to have her as a colleague at COA. We took the studies along the bottom. We took STURDY, Achieve, ERIC, NHATS, and BLSA. And we looked at frailty in each of them. And frailty is a huge problem for older adults. And physical activity is a big part of frailty, but it's really poorly measured. Historically, it's been very poorly measured as part of frailty. It's been self-report of various activities, which can be very hard to quantify and define accurately. So we want to take a deeper dive into physical activity and frailty and see what it tells us. So here again, you see these diurnal patterns by frailty status, the blue across all of these studies. So the blue is people who are frail, the yellow is people who's pre-frail, and the red is people who are robust. And I think what's important here is Obviously, those who are frail and pre-frail less active, but look at the overnight hours. So the early morning hours, we see here that there's the people who are frail are more active overnight. So this may be a sign that they're actually not sleeping as well, right? So activity at night is a sign of poor sleep. So sleep researchers say they're measuring sleep, but they're really measuring the absence of movement, right? If you're not moving around at night, the hope is that you're sleeping well. It's either that or you're not wearing it, but let's hope that it means that you're just not sleeping well. And when you quantify these differences in a scale that's easier to see, what that means is that at 4 a.m., frail people are about 40% more active than people who are robust. That's a big amount. Even if it's just a little bit of activity, that's a huge amount. 40% more active means they're not sleeping as well, which could mean a lot of things. It could mean that they have diseases that are manifesting and keeping them from sleeping well. It could mean they're at risk for disease. It could mean a lot of things. But then at 7 a.m., things reverse, and we see that the frail participants are about 30% less active. So they're more active overnight, less active during the day. This is a dangerous pattern. So where do we go from here? I like to think I'm just getting started. I like to think I have another 20 years of this, but we'll see. I think we need to continue in this vein that we've been in, is measuring health and function in novel ways, thinking outside the box. Moderate to vigorous physical activity is important. I'm not going to stand here and say it's not important, but it's not the only thing that matters. And we have these incredible tools, and we have this ability to collect this rich, dense data and really, really learn from it about how people function in their own environment. I think we need to figure out how to combine data across multiple sensors. So anybody who has one of these knows, oh, it measures my sleep, it's measuring my oxygenation, it's measuring all sorts of things. There are continuous glucose monitors. We have all these signals around us all the time, but we need people who are whizzes with the data to tell us how to combine these signals and understand what that actually means. We need more data in older people. So I'm certainly working on it. When I showed you the previous slide, we have about 10,000 older adults. But the problem is a lot of like our nationally representative data that we've had today, especially NHANES, is not that great in people over 60, and over 80, it's almost non-existent. So we don't know what's normal for a 60-year-old or a 70-year-old or an 80-year-old or a 90-year-old. And we need to understand that. Once we know what's normal, then there's clinical meaning. Then the doctor knows you're more active, you're less active, something needs to be done, something's not right. We need to define meaningful change in activity. So if I say you lost 1% of your daily activity, what does that mean? Does that matter? Is that a problem? Do I need to do something? We don't really know yet. What does that percentage decrease or the percentage decline mean clinically? We don't know yet. We want to quantify the healthcare cost of inactivity. So, and I think we're going to have the ability to do this in NHATS because we can link it to people's Medicare claims data and understand what is the cost of inactivity, because then it'll get more attention if we can demonstrate that people who are more active, people who are moving at different times, or people who are sleeping at different times actually cost less, then we'll get more attention. And I think we need to think about how to incorporate sensors into clinical practice for early detection and remote monitoring. So a lot of the work that we've done, especially on the cognitive side, is looking for differences in movement that could be indicators of poor things to come. The problem is a lot of times it's not specific, right? We just know something's not wrong, someone's not moving right, someone's not sleeping right. We might not know why, but if the clinician is working with the patient, hopefully they have an idea about their medical history, and that will give an indicator that something's off, something's not right. So with that, I want to thank a lot of people. I think I got everybody, but I'm sure I forgot somebody. People who have really helped me in all of these projects since day one. First and foremost, up in the left corner is Luigi Ferrucci, who I would not be up here if it weren't for him, his belief in me. He hired me when I was a new master's student, but also just his belief that I could do this. People here at Hopkins, most of them across the top row who helped mentor me as a student and as a junior faculty member. My NHATS colleagues over here in the corner, Vicki and Maureen, who I talk to almost every day about something that's going on in NHATS. My Epi of Aging contingencies in the second row. Some of them are former mentees that have become colleagues, which I'm absolutely thrilled about. Alden and Jennifer Deal, who are my academic siblings. We've been together for 20 years almost, which is really hard to believe. We came here at the same time and never left, really. And then a lot of other collaborators that I've just been so lucky to work with through the years, people at COA who work there now, students, staff, and also other faculty members. You guys are great. You make my job a pleasure in many ways. So thank you for that. My family, who is here in the front corner. So first off, my parents, who are in the upper left corner, but also here, who are 84 and 85 and rocking it. You guys make me look like I know what I'm doing. So thank you. You make me look good. And then my kids and my husband, you guys are amazing. You make every day, some days are tougher than others, and you guys make it worth it without a doubt. And then anybody who's been to my house or knows us knows we have a large furry contingency at our house, and some of them aren't even on here. The chickens didn't make it on here, but cats, dogs, sheep, donkeys, chickens. It's a really fun time. So if you ever want to come visit, just let us know. But you know, they make it all worthwhile. So thank you to you guys very, very, very much. And then, of course, I have to acknowledge all my funding through the years, because without that, none of this would have happened. And just another shot of us at GSA, Gerontological Society at COA Group a couple years ago. That was a super fun dinner. So thank you. Any questions?
Yeah, so that was absolutely fantastic. Really well done. And we do have some time for questions. So, and those of you online, if you'd like to ask a question, either raise your yellow hand, or you can put something in the chat and say you'd like to ask a question. We'd like to call on you so you can ask the question. And we have a couple of microphones. Wow, it's all crystal
Jen, that was
clear,
great.
Right?
Thank you so much. That was really interesting to follow all of the pieces that you put together. I have a question. Maybe not. Okay. I have a question about what your thoughts are regarding the idea that some of these signals might be signaling other comorbidity, right? So we've looked in different diseases, and you talked about the phenomenon of accelerated aging. And I'm wondering, you know, there's a little bit of it like a chicken and an egg with a lot of this.
Yeah.
So do you see the intervention point as increase the activity through structural intervention, individual level intervention, in order to ward everything off, or to look at it as kind of the canary in the coal mine for things that have yet to come? Like, where do you see the actionable information?
So that's a great question. So I've participated in 2 interventions since I've been here at Hopkins. I'm not an interventionist. I like to observe things. I like to see what's going on and try to put the puzzle together. Amal wants to be an interventionist, so I'm going to let him maybe I should let him answer the question. But the 2 interventions we've done, so Achieve, and I did show the hearing intervention did nothing in terms of physical activity. The other one was Sturdy, which was Larry Apple was the PI. It was vitamin D and falls. Neither one did anything, but they were both in 70 years and older. And even in some of the bigger trials that are not from here, like the Life trial was a big one that was done kind of, I would say, when was that? When were the primary results published? 2015, 2014? Any trials that we see in older adults, they work, like especially a structured physical activity trial, that works, but the problem is the sustainability. And if you wait until 70 years old, it's very, very hard to modify a lifetime of bad habits. It's not impossible. It's just really, really hard. And so I think what we need to do is think outside the box in terms of interventions, teaching people to be more active in their own environment rather than having them come to us 3 days a week and work out in a gym. Anybody who does that's going to be more healthy. But what are they going to do when the trial's over? Right? Are they going to still do it on their own? Are they going to find a way to do it on their own? We don't know. I think teaching people with wearables and things like that to know how much they move, or more importantly, how much they don't move, might be more motivating. As far as the other thing, like canary in a coal mine, I think it can be a signal, right? It can absolutely be a signal to physicians, to caretakers, caregivers, that something's not right. Person's not sleeping well, person's not as active, maybe they're up wandering at different hours of the day. Those kinds of things are things that we need to understand a little bit. It's a signal. Is it a specific signal? Is it telling me that person has cardiovascular disease? No, but it is telling me that something's off, right? And especially when we think about things like cognition and how that manifests, probably a lot of the pathology starts in midlife. We need to start thinking about how those changes might start early in the process. I can't tell you how many of these papers we write where it's like the pathology is already there and prevention needs to start earlier, right? And so I think we need to think about aging more broadly. I mean, aging starts arguably at birth. And so we need to think about, aging in that regard. Does that answer your question? Absolutely. Okay.
Jen, great job. You left out a lot of the work you've done on measurement.
Yeah,
You alluded to
ton.
it you've done a lot of work related to that. And you talk about your research quality devices and the rigorous methods that you and team apply for those. Yet most of the data that is out there on physical activity is from the Fitbit and from your Apple Watch. And
Right.
I'd like to hear your comments on how and Fitbit and Apple Watch's goal is market share, not on rigorous analysis or
Right.
necessarily providing high quality interpretations of these data. So
Yeah.
thoughts on how to translate your academic rigor into that commercial space
Yeah.
and how you might partner with them or advance your
I
thoughts.
don't know if they care, but no, it's a great question. So yeah, and Greg is right. I didn't talk about methods. A lot of what we do is around methods. You'll notice that when I present results, I don't call it steps per day. I don't call it MVPA in most cases. And there's a reason for that. And it's because there's a huge problem with misclassification and error and bias in that. When I taught the Epi Methods series, I used physical activity examples all the time because I thought students can probably relate to it. I have an Apple Watch. I like my Apple Watch. I look at it. I think it works pretty well, but there'll be times where I'm standing at my desk like this and it'll ding me to say stand. So there's measurement error in there, right? And people can observe that on their own. But understanding how they work in each person is very, very difficult. Everybody's gait is different. Like I said at the beginning, it's like a thumbprint. And so your gait speed might not be the same as my gait speed. Your stride length may not be the same as my stride length. So when you see steps per day, it's probably a good approximation. I think it's a powerful tool for monitoring day over day within person, right? Within the same person, this tells me how active or inactive I was. But across populations, I like to stay away from it. And one of the reasons is because you can't get what we call the raw data from these devices. They give you what they want to call it. And we as scientists like to dig deeper. And they don't want us to dig deeper because we'll find something wrong with it and we'll publish it and then they're going to lose $1 million. So it's interesting because some of the, or I should say more than 1 million, but some of the papers, the earlier papers where people did publish their algorithms, that's exactly what happened. These companies publish their algorithms and then scientists find something that's wrong with it and then they go out of business. So like Nike, the fuel band went out of business. I mean, the basis, the job, there's been so many that have gone out of business. Apple will not tell us and probably never will tell us how they developed their algorithm or what they used to do it. But I can almost guarantee you that they don't have a sample of 80 and 90-year-olds in there wearing them as well as the young, healthy people. And so I stay away from it. I stay away from it. Will they work with us? I don't know. Maybe it'd be nice, but they're not really beating down the door because they're profitable enough on their own. They don't really need us. So that's part of the problem. Yeah. Yes.
Any other questions?
Yeah.
Yeah, sure.
Amazing talk. I just had a bit of a follow-up question, I guess, in terms of measurement. There's like an active debate in terms of like sensors being sensitive to like color of skin. So I was just wondering if you've like sort of segregated the data in terms of race or anything like that?
That's a great question. So most of what I presented today was just activity. And so the sensors that measure activity are not sensitive to skin. The things that are sensitive to skin tones are if you have one of these and you turn it over, you see the little green flashing lights or the red flashing lights. It's called photoplethysmography, and it's how it measures blood flow, to get heart rate, to get oxygen saturation, all of these things. Those are sensitive to skin tone. So the research grade devices have lagged behind the commercial devices in measuring those kinds of things. And the reason why is because they want to get FDA approval. They know that scientists are going to validate them, things like that. They're at a higher standard. That being said, Actigraph, which is the main company we do business with for their sensors, has just started putting heart rate sensors on, and we put them on people in Eric. And at the Mississippi site in Jackson, Mississippi, where almost everyone is Black, we get 4 days of data versus 6 days of data in the white participants. And so the reason is it's not the accuracy, it's that the sensor has to work harder to get the same signal. So the battery runs dead quicker. So some sensors don't work as well. The higher grade sensors, they work. It's just they have to work harder. So the battery life is shorter. So we're trying to figure out what we're going to do about that. And we had the same thing. We are trying to do this internationally. We'll see what happens. Longitudinal study of LASI, longitudinal aging study of India. We're going to put them on people in India. We did pilot that. We got about 5 or 6 days in most people in India. So that worked pretty well. But in our lab, we have people of all different skin tones, and we see almost a linear association. The darker your skin tone, the less battery life you get. So yeah, it's a good question, and it is definitely an issue. So yeah.
We have time for 1 or 2 more questions. Lataska, anybody online? Anybody online want to ask a question?
It's okay if everyone just wants a snack. All right, too.
So with the current administration and their focus on making America healthy again, and how does all this work fit into their priorities? Do you have a sense of that?
I don't know yet. I would hope that it would fit in, right?
Yeah.
If we want to know how healthy people are. I mean, eating right and exercising are 2 of the biggest pieces of
Yep.
public health advice.
Yep.
But everybody knows they're supposed to do that, and we all have trouble doing it.
Right.
But maybe this kind of research can help support that.
Yeah.
Right? How important it is. And it doesn't have to be, MVPA, 150 minutes. I think the guidelines are part of the problem. I think the guidelines are confusing to people. People don't understand what's moderate to vigorous physical activity. How often do I have to do this? How hard does that mean I have to move? How fast do I have to walk? I think we need to make things easier. I think we need to leverage things like monitors so that people can understand if they're where they need to be, and if they're not happy with their health, then maybe they need to do a little bit more. Maybe they need to sleep a little bit more.
Mm-hmm.
I would hope this kind of thing would fit in with Make America Healthy Again, but I don't know.
Yeah.
Time will tell.
We'll wait and see. I think you have to get out there and make the case.
Yes, and aging too.
Yeah,
like you said,
absolutely.
We are
Yep.
aging population. We're an aging planet, and so understanding these kinds of things is tremendously important. So
Right.
Yeah.
Great.
Thank you.
All right. Well, again, thank you so much for a great talk, but all that you're doing. And you mentioned to me right before we started that you're going to be offering a course.
Yes. So
you want to give a plug for the new
we're
course?
teaching a course. Well, it's not till 4th term next year. So we have about a year.
Uh-huh.
So we have to roll it out a little less than a year. But I give lectures a lot of guest lectures in a lot of classes. So CVD, epi, nutritional epi, like a lot of different classes. And so there just always seems to be a tremendous response from the students. So, and interestingly, at the School of Public Health, there is not a single class on physical activity, which is so amazing to me. So we proposed one that was accepted. And so we're going to start that next March. So it'll be a lot of this and more measures, monitors.
With probably a lot to do with measurement as well.
Yeah.
Yeah. Great. Well, that'll be a terrific addition.
All right.
Well, again, thank you all very much for coming, both here in the room as well as those of you online. And a special thanks to the family. You are what made it happen, literally, right?
Literally.
But we all have a sense of gratitude towards our family, and they mean so much to us. And so thank you very, very much for all that you have done to make an incredible career happen. So thank you. So, well, thank you again very much for coming, and we'll invite you back in a few years and see where things go. So everybody, now's the time to get up, walk around, exercise a little.
Stand up.
Right. All right. Thank you.